If you like to cook, or in my case, eat, chances are you've got a cookbook lying somewhere at home. In any cookbook, there are plenty of recipes to choose from. Some of these recipes you might love and cook them often, whereas others not so much, you may never cook them at all. The cells in our body are surprisingly much the same. They all have the same cookbook, our genetic code, but vary in how they pick different recipes, except in their case, which genes they express. Now these genetic recipes have to be followed perfectly for healthy human function. So any changes to the recipe can cause serious medical side effects and conditions, such as cancer. Cancer continues to be a leading cause of death in Australia and worldwide. In 2020 alone, cancer accounted for approximately 50,000 deaths in Australia, whilst worldwide, cancer accounted for almost 10 million deaths in 2020. It's therefore clear that cancer is an issue that needs to be addressed. Now, although cancer cells come from the same cookbook as our regular cells, during their development they figure out that some recipes alert our immune system and get them killed. However, if they can learn to avoid these recipes, cancer cells can evade our immune system and continue to wreak havoc. This is a huge issue. Our immune system is the fundamental defence we have in preventing cancers from arising and progressing. Therefore, the fact that cancer cells can evade our immune system poses a major hurdle to cancer therapy. So what can we do about it? Well, my research involves the development of a novel cancer therapy using state-of-the-art genetic editing technology called CRISPR to force cancer cells to cook the recipes they try to avoid. That is to say that we make them read the immune alerting genes. This technology has been already applied successfully in clinical trials to improve the killing capacity of patient immune cells. But in my research, I aim to instead target the cancer cells directly and make them more receptive to therapy. Using breast cancer as a model, I've been able to show that this genetic editing technology can activate a wide range of immune genes. Now, my job is to identify which genes and which combination of these genes best improves the capacity of a breast cancer cell to be eliminated. Given the high rates of relapse and drug resistance seen in current cancer therapy, we believe that a targeted therapy such as this can be a new treatment alternative for patients. Moreover, although my project focuses on breast cancer, I firmly believe that the genes we identify can be translatable to other cancers also, potentially allowing this treatment to benefit a wide variety of immune genes. In the long term, we hope to develop a novel, effective targeted therapy that acts both independently and in combination with currently available therapies. With over 150,000 cases of cancer predicted to occur this year alone, the time for novel and effective therapies is now. Thank you.